Hey folks, welcome to your first ever AP Biology flipped lesson. This is going to be the pre-lesson to see if you're actually into this kind of style. I don't mind spending the time if this is a resource that would be helpful for you. What we wanted to go do is get you a little bit familiar with the AP Biology site, as well as familiar with the AP Biology course and exam description. It's also called what we say the CED, course exam description. Basically, it's the Bible for the whole entire course. It tells us everything we're going to go need. The link of it is already located on Schoology. We'll show you where that is. If you don't remember, it's in the main course information, the introductory stuff. So I'm going to go ahead and click this guy. This is a huge document, right? We can go take a look and look at the contents and stuff like that. Talks a little bit about what the class is about, the framework and stuff, and goes into a lot of detail about the different units, right? So remember me doing my spiel of like, oh, unit 1 is chemistry of life, and then you got like cells and stuff, right? So you'll see that everything that you need to be aware of is located in here. Anything that I design my lessons, my labs on, I'm using this in order to go ahead and construct it. It basically provides the framework for the whole entire class. What we're going to go ahead and do is scroll through this guy. Right? We're going to go ahead and get down to really the important stuff on how the class actually ends up working, specifically in the framework. Okay. What we want to go do is talk about these science practices. Okay. How this course is designed is that there are going to be 6 practices that we're going to go play around with. And I already mentioned them in class on the first day. It's basically being able to describe what's going on, right? Then being able to model, right? And eventually it's going to go and make its way down to being able to form an argument, right? The idea on how this ends up working is that it's going to be built up like a building, right? You need a good foundation. You need to be able to describe everything that we're talking about in class before I can ask you to go draw a picture of it, right? And then after you're able to go and draw the picture of it, right, then we can maybe design an experiment, get data, get information, and then you'll be able to form an argument and really know your stuff for the course. Page 21 actually has the science practices as well as what goes along within it. So we'll just go ahead and knock through them real fast. Not going to go anything crazy, kind of show where it shows up actually in the document itself, the CED. So the first one is concept explanation. In the case here, you are literally what it's saying. You're explaining the concept. You can describe, this is being able to identify vocabulary, being able to explain what it is that we are taking a look at and what things specifically do, right? If we end up seeing animal behavior, well, in nature, right? Why is it that the animal behaves a specific way that it does? Why is it that the animal will be moving around randomly as opposed to moving in a straight direction? And well, we end up talking about this thing like called taxis and stuff, where an organism has a better chance of finding shelter. This is like a leaf right there. It's a better shelter in there, right? So that's all it is. We're not getting the nitty-gritty great deal. We're just saying that the first idea is concept explanation. Just tell me about what it is, right? Be able to feel good and be able to explain what it is we're looking at. Let's do another one in another color. In the case of visual representation, this is the modeling part. Can you create a model? If you can describe something, right? I say that the parts of a cell. You have the cell membrane, which acts as a barrier. You have the nucleus, which is the control center. It's where all the genes are stored in order to go and wait for them to be transcribed and eventually be translated into a protein. And I say, okay, cool. Draw me a picture of what a cell looks like with all these different components and visual representation, right? So here it is. You like be able to draw your cell with your nucleus, your little mitochondria in there, be able to go and draw some other stuff, like maybe like the endoplasmic reticulum. So you're able to draw stuff, explain relationships between it, right? So, okay, why is it that the endoplasmic reticulum is located so gosh darn close to the nucleus, right? Oh, hey, well, because the endoplasmic reticulum has ribosomes on it, it's a closer spot to go ahead and find proteins. So that way we can go and translate the genes even faster. Sounds good. Sometimes there'll be a picture and they'll give you some kind of like diagram, maybe a cell communication process. So we'll have like a receptor protein here and like some like ligand right there and some intermolecular stuff happening here with like proteins and other nonsense that, okay, cool. Well, this ligand attached to the receptor makes everything happen inside the cell. What would happen? We end up getting rid of the ligand, right? That's kind of like the stuff with visual representation. So the idea is that if you have a good foundation of what it is that you are looking at, that means that you should be able to look at a picture and be able to identify all the different parts to that picture. The next one, let's go do this one like a pink color or something. The next one that you end up having here is going to be questions and methods. This is basically doing an experimental setup is the way I kind of describe it to students. Can you set up an experiment in a way that it makes sense, right? Are you able to create the null hypothesis and then your experimental hypothesis, right? So case here, the null hypothesis that it's being correct, that means that your experiment was botched. You had to go figure out what is actually like the issue there. Can you identify your different variables? Are you able to identify independent and dependent variables? Can you figure out your controls and why the controls are put into place? It's funny, a little bit of a side. My wife is going for her doctorates, and she still like wasn't able to tell me the difference between the dependent, independent variable. And she's showed it to her professor. It's like, oh wow, you're like one of the few teachers, people who actually understand it. She's like, my husband's a science teacher. He just did it for me. It's just, it's just kind of a fun little joke in there. But yeah, it was pretty good. Let's go ahead and move on to the next practices, right? So if you have the ability to um, explain a concept, if you have the ability to create a model, then create an experiment or understand experimental setups, you should be good to go. 4, 5, and 6 is a little bit more lengthy. 4 is actually easy. All right, I'm going to go ahead and knock out 4 real fast. 4 says representing and describing data. It's a really funky, fancy way of saying, hey, can you graph? And do you know if you need a bar graph or a line graph? Right? That's all it is. And then being able like to read like, oh, what is the point at this area right here? What value should we have right there? What do you notice about the trend? Very, really simple. I think everyone can do graphing and looking at a data table and putting that on there. There's going to be lots of different types of graphs that you're going to have to be able to identify. We'll spend some time in class understanding, the differences between, like a box and whisker. Why would we end up having a pie chart in there? What the heck is a histogram in there? And we'll get around to that. Okay. The next one that we're going to go take a look at is going to be 5. 5 is statistical tests and data analysis. Hey, can you do math? Do you understand how equations end up working? That's really what the statistical tests are going to go and be. We'll show you the equation sheet at the end of the CED. You'll get this you'll get this equation sheet every single exam in my class. You'll get it for the AP exam. So do you know when to apply these equations when you need to? Do you understand how to figure out statistical analysis error bars, right? Being able to figure out your standard error. We like statistically significant data, which means that it's 95% certain. You may not know what that means. When we hit our statistics section in the beginning, with which we're going to go do with properties of water, we'll show you that. Being able to reject or accept the null hypothesis, basically meaning that if your null hypothesis was proven correct, again, your experiment was just botched or didn't end up, you weren't focusing on the right variable for what should have happened. All right, we got one more practice on top of this, and then we're done with science practices, which is argumentation. The idea here is that can you make a claim and then basically support it with evidence and stuff like that. And guess what? Can you use one to do concept explanation, right? Can you explain what's happening? Can you draw a picture or see how a picture relates to your argument? Can you set up an experiment or say that this experiment wasn't right because it lacked a control group or something like that? Can you do your graphs? Can you represent it, or can you do your statistical tests? That's basically what argumentation is. It's using all these other skills that we were talking about in order to go and form an argument, right? Make a prediction, right? So it says make a prediction if you were to disrupt something. Well, if you can end up using these ideas, right? Your model, like, hey, looking at the model, if I were to change this, this would not work anymore. And that's pretty much it for the science practices, right? The more that you see these practices, the better it's going to go ahead and be for you, and you'll be able to go and identify trends as well as things we'll see within the class. This right here is just showing you how the exam is going to be broken down. Unit 1, Chemistry Life, is 8 to 11%. Cell structure and function, 10 to 13%. The big heavy hitters is ecology and natural selection. That's going to make up about 1/3 of the exam, as well as cell communication as well. So if you'll see chemistry of life, we will spend a bit of time on it, again, for learning the mechanisms. It will show up probably for, protein chain sequencing and understanding how changing that could potentially affect the function. These questions, it's so much content and there's so much stuff to draw from. The course at a glance, I have this as a poster within the classroom. It'll be up, and what you can see is that it talks about what are the main skills that should be hit in each section, as well as all the different concepts in there. So if we remember from our science practices, you tell me, right, 2, 2, and 2, right? All these guys right there, which science practice are we really taking a look at, right? This is going to be modeling. So the science practice that we're looking at here is making sure that you can do visual representations for structure of water and hydrogen bonding, right? Elements of life, can you show these nutrient cycles? Introduction of biological macromolecules, can you explain to me how dehydration, synthesis, and hydrolysis works with using a picture, right? While this one right here, properties of macromolecules, this is all focusing on concept explanation. Go ahead and explain to me why or what they end up doing, right? What does a protein do? What does a lipid do? What does a carb do? After you go into the properties, then we go into the structure and notice that, right? It says argumenting, right? Structure and function. Why is it that the order of amino acids determines the function of what the protein ends up doing, right? Well, an amino acids will fold in a specific way, and you change how they want to fold, and it's not going to fold exactly the same way. We're going to go show you that stuff. You can take a look at all the different courses or all the different sections that are in here. Every single one will have different things they want to go ahead and highlight. You'll see lots of 6s all over the place, right? Because we want to make sure you're able to go ahead and argue lots of stuff. I wanted to bring this to your attention is that when we get around to it, natural selection is a monster of a class, or a monster of a unit that we're going to go get to eventually. But you can go take a look at the diagram, the stuff, and you'll be good to go. Before we concluded this little video, what I wanted to do is get to the equation sheet. So I wanted us to actually be able to see the equation sheet as you would get it. So it should be in the back here. It should be like an index region. Let's see here. Where are you? My little equation sheet. It's like probably right here, back at the bottom. There we go. This is the equation sheet. I'm going to make it a little bit bigger and stuff, so that way it's a little bit easier to see. All right. AP Biology, equations and formulas. Everything that you're going to go need for any math related thing in the class is going to be located right here. It says standard deviation, mean, standard error of mean, chi-square. We'll show you when all these are going to end up being applicable. Instead of just doing a whole huge math lesson, it's a lot better for us to just use it when we need it. To kind of give you an idea, scroll too far with that. Standard deviation can be used in any experiment where we have multiple samples and try to figure out how our data is different. Standard error of mean is figuring out a little bit more of how much your data is different by. Chi-square is going to be used to see if testing for null hypothesis, alternative hypotheses. Basically, we'll go into genetics, we'll breed some kind of organism, right? And we're expecting it to be like a 3-to-1 ratio, 3 dominant and 1 recessive. And you look at results, they're all recessive, right? It basically would plug that into this equation, and then we'd use this degrees of freedom chart right there to see if it's above or below. If it's below, you're fine. If it's above, it's not good. Probability is goes along with genetics. I've never had to go use that. Hardy-Weinberg is specifically for evolution. Um, we go down further. Rate and growth, this is all for ecology. Water potential is the bane of a lot of students' existence when it comes down to why water works the way that it does. This is specifically in osmosis. Simpson's diversity index is how to determine if an ecosystem is healthy. And then you got surface area to volume ratio, which is for cell size. So we'll be seeing this a lot in cells and genetics, as well as finding this in evolution and ecology. So math is sprinkled in when it's relevant and be good. All right, that's going to go ahead and conclude this. We'll reconvene and start talking in class.